Okay, so we're going to look at factors that affect the way particles move across the cell membrane, and we're going to talk about two general factors that influence the rate. All right, some factors that influence the rate of how fast those particles are moving across the membrane. Okay. So the first thing we'll look at is the rate of transport. Now, we really want to understand here that particles aren't moving at the same rate all the time. Right, so particles will start to move freely if there's a really large concentration gradient, and then eventually they'll get to what's called diffusional equilibrium. And we kind of talked about that in the previous packet, right? That's where particles want to be. That's what the chemical driving force is trying to achieve. Getting these particles to where they're no longer moving, they're at equilibrium, okay? And so what we see here in the first scenario is we have a lot of movement of the particles. So there's a force going in this way, there's a force going in that way, and so the net flux, and we talk about net flux, that's the total accumulation of particles on one side or the other, or the total movement of particles to one side or the other. So here we have particles moving freely, where we do have a net flux going to the right, and then particles will eventually get to looking like to this at the top here. Okay, where we have equal concentrations on both sides of the membrane. And although we still have individual particle movement, right, it's important to understand here that even at diffusional equilibrium, particles are still moving in this top scenario here. But there's no net accumulation on either side. Okay, and so there's no net flux. Flux is the total net accumulation on one side or the other. And why is there no longer a net accumulation in this top scenario? What do we no longer have? A gradient. We no longer have a gradient, right? Particles are still moving in either direction, as we can see, right? There's a one-way flux, but there's no net flux. We have no gradient, okay? And so these are the two sort of ends of the spectrum of the rate of transport across the membrane. We can go freely moving, or we can get to equilibrium where we're not moving much at all, right? Two ends of the spectrum. All right, so the first mechanisms we'll talk about are passive transport. We're going to look at three individual mechanisms under this heading. We'll talk about simple diffusion, we'll talk about facilitated diffusion, which requires a carrier protein, and we'll talk about channels, which is somewhat similar to facilitated diffusion. Now, we just want to remind ourselves of the features of passive transport. It is spontaneous, it requires no energy, and it's a downhill type of movement, okay? It's actually going to generate energy. We'll see later on that passive transport actually generates energy to fuel some of the active transport mechanisms that we'll also talk about. All right, so these are the three types that we're going to discuss. The first one being simple diffusion. Now, we want to understand here that there are no membrane proteins required. And this simply means that there's no facilitation required whatsoever. There's no mediators, there's no carriers required. These particles are moving freely through the lipid bilayer, okay? And so there are three factors that affect how fast these particles will move. The first one is the magnitude of the driving force. And when we talk about the driving force with regard to simple diffusion, we're usually talking about the chemical driving force. We can include the electrical driving force, but we're usually talking about that concentration gradient, right? If there's a bigger concentration gradient, particles are going to move faster to get to equilibrium. If there's a smaller concentration gradient, particles are going to not move so fast until the particles get to diffusional equilibrium, where they're not moving, they're not net moving in either direction. The second factor is the membrane surface area. We'll talk about some features there. And then the membrane permeability. How easily do these particles move through the membrane organically? Are they built, are they structured to go through that non-polar center of the lipid bilayer? All right, so let's look at an example of the magnitude of the driving force and how this influences the rate. All right, so we're talking about simple, We're talking about simple diffusion here. All right? And we're basically saying that the magnitude of the driving force, and again, most commonly we're talking about the chemical driving force. The magnitude of the driving force determines the rate of simple diffusion. Okay? And so let's look at a scenario. We have 3 different cells down here. All right, let's call this cell A, cell B, cell C. These cells are separated by a membrane going right down the middle. And we have varying concentration of solutes or particles on either side. So in this first cell A, we have 1 mole of particles. And that basically means we have some solutes dissolved here. Whereas we have 0 moles here, and this is basically no solutes, so that's pure water, okay? In this second cell here, we have 0.8 moles, and then we have 0.5 moles, so slightly less. And then in C, we have the same concentration of solutes on both sides, okay? All right, so what we'll see here naturally is that in cell A, in this first scenario, we have a much stronger force here, right? And the reason we have a stronger force, as well as a stronger rate of movement across this membrane, is because we have a strong gradient. We have a strong chemical driving force, right? So a strong chemical driving force. That gives us a faster rate, right? In scenario B, we have 0.8 moles on the left and 0.5 moles on the right, so we have less of a gradient, right? It's not as strong a gradient as this first scenario. So we have sort of a, let's say, a medium chemical driving force, and so we have a medium rate, okay? Whereas in this final scenario, do we have a gradient here? We don't, right? The concentration, the amount of solutes on either side of the membrane is the same. And so while on this side, while on this scenario, we had a weaker movement, on this final, in this final cell, in this final scenario, we have movement taking place in both directions, but this cell is at diffusional equilibrium. I cannot say that word. Diffusional equilibrium, and so there's movement of individual particles taking place in both directions, but no net accumulation on either side, right? So we have no chemical driving force here because we have no gradient, right? And our particles are at equilibrium. Bless you. Our particles are at diffusional equilibrium. Definitely got to sound that word out. All right. So this really illustrates to us that we can have a strong rate that is influenced by a strong driving force that is influenced by a strong gradient, right? And then we can have a weak rate, or actually no movement, right? These are at equilibrium because we have no driving force, because we have no gradient, okay? All right, and that's simple diffusion. That's the rate of simple diffusion. Okay. So again, looking at the magnitude of the driving force, the chemical driving force, we can plot this on a graph, okay? And if we plot the net flux, again, the net flux is just that net accumulation, that net movement to either side, and we plot that against the concentration gradient, we can see that there's a directly proportional relationship, right? This is an indefinite directly proportional relationship. As long as the concentration gradient increases, so too will the net flux increase, right? The bigger the concentration gradient, the bigger the rate of movement, the bigger that net flux. Okay, that was the first factor that we said influenced the rate. The second factor was the membrane surface area. Now, the amount of surface area that's exposed will determine how fast those particles move across the membrane, right? Think about it. If you had, in the case of the lungs, this is an image of the alveoli of the lungs, and on the left, we can see a normal patient, right? A normal anatomy of the lungs here. This is an electron micrograph. Whereas on the right, we can see that we've lost some of that surface area. Right, and this happens with lung disease, and we lose the surface area, so we have less of that oxygenation taking place. And so what this really underscores to us is that the more surface area exposed, the more movement, right, the faster movement across that actual membrane. Right? Think about the large intestines. Think about, again, the alveoli of the lungs. We have to create structures. So for example, the villi of the intestines is intentionally highly folded so that we can really create that surface area to have movement of nutrients from the gut into the underlying blood vessels. And then the final factor that influences the rate here is basically the membrane permeability. How likely are these particles to go through the membrane? And the most important factor that influences permeability is obviously the lipid solubility. We talked about this when we looked at the structure of the membrane. Because the membrane is a lipid bilayer, it's made of those amphipathic phospholipids. Certain particles are not going to go through the membrane. Water, for the most part, will not go through the membrane. Charged particles will not go through the membrane. Larger, more bulky particles will not go through the membrane. Okay? Nonpolar, hydrophobic particles such as your fatty acids, fat-soluble vitamins, steroid hormones, those things will easily move through the membrane. Second here is the size and shape of the particle. Now, bigger, bulkier, irregularly shaped proteins are going to go through the membrane much slower than smaller particles, smaller non-charged particles. Carbon dioxide, oxygen go through the membrane much faster than some of our bigger, bulkier molecules. And then finally, temperature. Now, we have here that temperature is not really relevant in human physiology because we maintain such a tight, a narrow window in terms of our temperature, our core body temperature. But in terms of outside of the body physiology, temperature makes molecules go faster. So they're going to be moving at a faster rate, and hence they're going to move across that membrane at a much faster rate. So that becomes important when we're looking at transport outside of human physiology. And then lastly, the thickness of the membrane. This just sort of makes sense, right? The thicker the membrane is, the slower we're going to have movement of particles through that membrane. There's just more stuff to get through. So a particle is going to move at a much slower rate through a thick membrane than it will moving through a much thinner membrane. And this is why some of the body structures, some of those membranes are designed the way they are. That's why the alveoli are very thin, right? That simple squamous epithelium, that very simple epithelium that'll allow the movement of particles through very easily. Okay, so the thickness of the membrane is really important in determining the rate of diffusion.